Today we will discuss how genetics will kill fossil fuels. Biofuels use plants, algae, and cyanobacteria to convert CO2 and sunlight into usable forms of fuels. These fuels are usually sugars, fats, and alcohols. Sugar is extremely energy dense. One sugar packet contains about 6 AA batteries worth of energy. Another fuel created by biofuels is fat. 4 grams of fat contains about 14 AA batteries worth of energy.
Cyanobacteria strains are the most promising for fuel generation. Cyanobacteria is a primitive prokaryotic and multicellular organism capable of photosynthesis. One byproduct of this photosynthesis is ammonia fuel production. Cyanobacteria were heavily present during both the Archean and Proterozoic eons. When the cyanobacteria died, they fell to the surface forming stromatolites. A large portion of currently extracted crude oil originates from these decayed cyanobacteria. However, we can also use the living descendants that created this ancient power source to harvest fuels today. The heterocyst on a cyanobacteria is a modified cell that produces ammonia. Ammonia has been occasionally used in the past as fuel for internal combustion engines and fuel cells. Ammonia combustion also does not release carbon as a byproduct and has a high octane number, which withstands compression before detonation better. However, the production of these biofuels could be enhanced using CRISPR. If we have a target gene which we wish to eliminate, we can create a complementary guide RNA strand which matches the genetic code of the target. This allows us to identify the gene we wish to eliminate. We can then attach an enzyme known as Cas9. The Cas9 will act as the cutting mechanism, allowing us to cut away the target gene. If the guide RNA matches the target gene, the Cas9 will cut away the target gene, eliminating it from the genetic information of the cell.
One of the most promising bacteria is Cyanobacterium Synechococcus elongatus.
This cyanobacteria was optimum for biofuel production due to its rapid growth rate. They target a gene called NBLA. They targeted this gene using CRISPR-Cas9 due to the gene playing a role in breaking down phycobilisomes. Phycobilisomes are responsible for photosystem II, which is a stage of photosynthesis. Thus, by preventing phycobilisome breakdown, this leads to more phycobilisomes. As a result, the cyanobacteria experienced hyperactive levels of photosynthesis and thus entered fuel generation. Algae is also a very promising fuel alternative. Algae has CO2 sequestration properties, fuel production abilities, and is prime for CRISPR enhancements. Algae can be used to lower atmospheric CO2. Already, 45% of our planet's carbon dioxide absorption is facilitated by algae alone. Furthermore, some companies have begun planting algae farms next to their fossil fuel plants to aid in carbon dioxide sequestration. In fact, doing so reduced carbon dioxide emissions from these facilities by 80%. For example, an algae harvesting plant built by Global Algae Innovations' Kauai Algae Farm in Hawaii set up right next to a fossil fuel power plant. This plant successfully reports helping to extract 80% of the fuel plant's CO2 emissions. The high carbon dioxide environment is also mutually beneficial for the algae growth as the algae feeds off of the carbon dioxide as a nutrient source. Green Plains Renewable Energy and Bioprocess H2O Team-Up have similarly teamed up in a project to reduce waste from a corn ethanol plant. Together, they are using the CO2 wastewater and heat byproducts from the corn ethanol plant to supply nutrients for genetically optimized algae growth. When algae is combined with a catalyst, a process known as transesterification is facilitated. This is when fat divides out of the algae cell. After purification, the main fuels produced include biodiesel and jet fuel. If we allocated 0.42% of land to algae fuel production, we could supply 100% of our country's energy needs. That is around 10 million acres. For comparison, corn takes 92 million acres. Even this, however, can be reduced due to new growing techniques. Algae growing is typically done using very large ponds. However, Green Plains Renewable Energy has developed a vertical algae growing system for genetically modified algae better suited for surviving in this new environment. In doing so, they have reduced land usage for animal feed production by 40%. Typically, most farm plants grow about 7 tons per acre. For this modified algae, a vertical farm could harvest 15 to 40 tons per acre. They can also harvest the algae several times per week instead of once per year. Current algae growing techniques include vertical tubes, horizontal tubes, raceway ponds, suspended cord vertical farms, and vertical sheep farms. Some limitations of using algae as a primary biofuel source include the high production costs associated with production and the high nitrogen needs of algae. Production price was 10 times too expensive and since has been mitigated in recent years using pressurization to speed up the transesterification process and by using algae genetically modified for optimized vertical growing platforms to reduce land usage. Global Algae Innovations' Kauai Algae Farm has reduced costs to around 1/2 that of crude oil. The algae is also highly nitrogen dependent. Providing the necessary nitrogen-rich environment can be difficult and costly. CRISPR has enabled us to lower nitrogen dependency. Using CRISPR, scientists were able to identify 20 genes that control the suppression of fat production within algae cells. They were able to use the Cas9 enzyme to delete these 20 genes, and as a result, the production of lipids, which is the primary fuel source derived from algae harvesting, was doubled. Using this CRISPR technique, Synthetic Genomics has said they will be able to make 10,000 barrels of algae biofuel a day by 2025. Synthetic Genomics first grows their new CRISPR genetically modified algae strains in test tubes. They then test the growth of the modified algae specimen on petri dishes. If the stains show promising signs of growth and conversion of carbon dioxide to energy-rich fats, they move the algae to large-scale growing chambers that provide optimum growing conditions. After a sufficient amount has been cultivated in the growing chambers, they then move the algae to greenhouses containing scaled-down raceway ponds. Here they further test the algae's ability to survive. If the algae grow well here, they move to the large-scale outdoor raceway ponds.
8 to 10% of our total oil supply goes to making plastics. This comes to around 9 million barrels of oil per day. What is the solution? Bioplastics could be used to replace the current use of petroleum to create plastic products. They ferment sugar saccharides and produce polyester materials. Yeast and bacteria are used to create polymers that mimic the physical characteristics of plastics. Currently, bioplastic production is done using corn, wood shavings, vegetable oils, and food waste. Senecocystis and Senecococcus are cyanobacteria species that are good for bioplastic production. This is because under nitrogen and phosphorus starvation, they can produce polyester materials. Genetic manipulation has succeeded in increasing the productivity of polyester materials, polyhydroxybutyrate, succinate, and lactate from these cyanobacteria. Monsanto acquired Biopol from Zenka in 1996. Monsanto is creating bioplastics from plants instead of microbes and bacteria. Monsanto genetically modified rapeseed by adding 4 genes from a bacteria that naturally produced biodegradable plastics called PHBV, thus creating a plant that can produce bioplastics. Genetic modification of plants using CRISPR will allow us to create cheaper biofuel products for plastics, reducing our crude oil dependency. Bioluminescence is also a rapidly advancing field of genetics that will likely reduce our dependency on fossil fuels. While bioluminescence gene editing technology has currently been applied to medical uses, future technology may allow it to be applied for home lighting and street lighting. Luciferase is the most commonly used bioluminescent. It is sensitive to a specific enzyme, and when reacting, it produces a green to yellow light. This results in an initial burst of light that decays over about 15 seconds to a low level of sustained luminescence. The gene encoding for firefly luminescence is known as the Luke reporter gene. What is beneficial about the Luke gene is that it does not require any post-translational modifications. This enables the gene to be better expressed after transplantation, making it good for gene modification. The reaction between luciferin and luciferase occurs when luciferin is subjected to the presence of oxygen. When the enzyme luciferase is introduced, the reaction is accelerated, releasing the chemical oxyluciferin and visible light photons. Another gene that has been targeted for use is the green fluorescent protein, GFP, naturally found in jellyfish DNA. This gene has been widely used in the medical field. GFP gene has been successfully inserted into human stem cells using a method called the safe harboring target. This is essentially where gene insertion is done by targeting a specific area in the genetic code where the cell will uptake the genetic information well. This location is known as the safe harbor. In humans, this site is called the AAVS1 site on chromosome number 19. If this type of genetic insertion can be done in humans, it might also be able to be done in plants if we can find the safe harboring sites. U.S. households consumed an average of 1,105 kilowatt hours of electricity for lighting in 2015. If bioluminescence were used for home lighting, we could decrease household energy consumption by 10%. Street lighting is often the first or second largest local government energy use, typically accounting for 20 to 50% of a municipal energy bill. CRISPR has also allowed us to insert the genes encoded for bioluminescence into plants. In 2013, Illumin, a plant-growing Kickstarter campaign, raised $480,000 to insert genes into a plant to make it glow, managing to produce a plant with a faint glow. Illumin admitted that the gene insertion process was actually a lot harder than anticipated. An MIT team also managed to implant nanoparticles inside of a watercress plant. The plant glowed faintly for about 4 hours. A French company named Glowy also made lamps using a gene from a Hawaiian squid. Their lamps claim to lower power usage by 20%. However, they only last about 2 to 3 days. There are numerous disadvantages to using bioluminescence to replace lights. Currently, the technology is very dim. It needs to be brighter for street lighting safety and for desirable levels of home lighting. For desirable home lighting, the colors also need to be modified. Currently, only blue and green light are expressed due to the genes only expressing for the production of specific light-emitting chemicals. Maintaining the plants can also be difficult due to watering and care needs, adding expense and time.